Hi, good morning. I hope you're doing good.
Today, I'll be talking about a key skill in bioinformatics, which is called spatial transcriptomics, or experts call it as a spatially resolved transcriptomics. It's not like RNA sequencing, but it's something even more advanced techniques than RNA sequencing technique. It can measure the gene expression from your cells within the intact tissue. It doesn't mix it up. RNA-seq, they take all the cells together and try to predict the expression levels based on all the mix-up happenings in RNA sequencing. But in spatial transcriptomic, if you have a tissue, if you consider a tissue and you want to measure the expression of each gene particular cells in that intact tissue, then you can define where the cells are coming from and what their roles in that particular tissue. It's completely different than RNA sequencing data. So if you would like to explain to a 5-year-old kid, then how do you explain that? So for instance, you take a big painting like a scenery with mountains, flowers, house, grass, etc. Each of the paintings has different colors, like grass is green, flowers are red, and house is brown. And in RNA-seq, what happens is if you mix all of them together, it becomes a mess. It becomes a brown mess, and you can't even recognize what color is the flower and what color is the house, what color is the mountain, what color is the grass, etc. But in spatial transcriptomics, that's the beauty of spatial transcriptomics, that the colors are intact where they are. Even if you mix it up, in that particular painting, they are like separated each other based on the colors. Like the flowers will be red in color, the grass will be green in color. Even though they are mixed it up, they will be categorized into different colors, so that you can track the object what color they are. So that's what spatial transcriptomics is, and it's a really important technique. And I mean, you can exactly say what particular cell is doing what type of activity and what is the expression level of that cell in that particular region of interest or area of interest. That's what a spatial transcriptomics is. It's a very powerful technique, and I think this is one of the skills that any of you, like a bioinformatician or any researcher doing next generation sequencing data analysis, they should know more about this spatial transcriptomics. Let's dive in. If you visit the 10x Genomics datasets website, you have all these datasets where you can choose the platform. So I'm taking the Visium Spatial platform. Previously in my single cell tutorial, I have taken this Chromium single cell. In this tutorial, we are going to explore the spatial datasets. So once you choose that, you choose any sort of species like human and mouse. In my case, I'm choosing the mouse. Okay. And sample and tissue, you can choose whatever you like. In my case, I'm choosing the brain. And I'm taking this particular data set, which is mouse brain sectional coronal. This is a spatial gene expression data set. If you open this, you have the summary about the data set, and you can see all the processed data sets from the FASTQ file. And these are the input files, which are FASTQs and the images of the spatially resolved imaging data. So this is the dataset overview, and this is what the image looks like. As you can see, each dot represents a cell, and this is the tissue that we'll be focusing on. You can read about the summary of the dataset. This particular dataset has been pre-processed using the cell ranger technique, and you can find the command over here, and you can see the analysis also, where you have different types of clusters, and also you have the expression levels they have analyzed already. But how you can analyze, that's the main goal of this video, how you can load this particular data set and how you can analyze the data set by using spatial transcriptomics. So from here you can download the data set, whichever the data set you like, and then you can load it to the Jupyter environment or the Python environment, and then you can analyze. Okay, let me also explain a little bit about the spatial omics tutorial, how you can do that. So like I'll be explaining an expert level as well as the 5-year-old kid, how you can explain to a 5-year-old kid. Okay, so we'll be using different packages like Scanpy, Squidpy, and some interactive visualizations like Plotly and Napari. And we'll be loading the Visium dataset from here, and then we'll be doing the pre-processing and the normalization using the Surat v3 flavor, because this is much more helpful. I mean, this particular flavor of normalizing the data to find out highly variable genes. And then we'll be doing the PCA, the neighbors, UMAP, latent clustering, and we have some static spatial plots and interactive plots with the Plotly and Napari. If you can explain to a 5-year-old kid, imagine we are mapping a city full of houses, each house with different lights, okay, and they are called the genes, and we'll color the houses by the light patterns to find out the neighborhoods and see which lights are on. Okay, because this is all about the genes on and off, you know, the expression levels which are up and down, which are on and off. Okay, so first thing what you have to do is you have to load the data set, and we'll be using this example data set that I have downloaded from here, and we can use the same data set as an example to load the data set. Then we do the pre-processing. So we'll be converting the data sets into a variable called a data, you know, the Scanpy normal process which converts the data sets into a data. So if you have to explain to a 5-year-old kid, then it will be like a data is our city map showing every house and its lights. It's like rows and columns with you know, in terms of genes, it's samples versus the genes. Okay. And then we do the pre-processing, normalize the gene counts, log transform the data, select only highly variable genes, and then scale the values so no single gene dominates. You know, we have to scale the values. So how do you explain to a 5-year-old kid? So it should be like, make sure all houses have fair number of lights, squeeze the brightness so huge lights don't scare us, and pick the lights that change a lot from house to house, and make all lights fair size to compare easily. It has to be uniform so that you can find out which are on and off, you know, like which genes are regulated and which genes are off regulated. Okay, and then we move on to PCA, neighbors, UMAP, clustering. Okay, and how do you explain that? So we shrink all the lights into few main colors, find houses with the similar lights, draw a map showing the neighborhoods, colors, houses in the clusters, and then you visualize using the UMAP where you can map the city with the neighborhoods colored differently. Sorry, green lights will have a different neighborhood, red lights will be different, and then we have some static spatial plots. Okay, so take out those houses back onto the city, the real city map, and color them by the neighborhood and the light patterns. Actual tissue coordinates can color by cluster or by gene expression, etc. And then you have the enrichment. Find the cells which are closer to each other. It's like a k-means clustering. So you cluster them based on the cell types and definitions. Each neighborhoods has a similar type of colors, and they live next to each other. And then you plot all of them interactively with the Plotly, and you can click each one of the cells and you can identify what these cell types are and how they are regulated, what are the nearby cells, etc. It's like the magic map where you can click the houses to see which lights are on. And then you have this Napari visualization. This is optional, but it's really helpful those who are doing spatial transcriptomics. So you have to identify the colors just by visually looking into that, because in RNA expression analysis you have all those volcano plots and all different types of 2D plots, but here you can actually see from that particular tissue, like this one, where the cells are, what type of cells they are, and how you can visualize what they are what the activity is, what is the expression levels. So let's dive into the notebook, and I will explain you step by step same operations as a spatial transcriptomics datasets from the 10x Genomics, and I will perform the whole analysis over here. Let me show you the requirements file where you can install all these required packages using the Python environment. You can install this particular requirements.txt file as pip install -r requirements.txt, and I have listed all of them here that I have used to load my notebook and to run efficiently all the pipelines using the Scanpy and the Squidpy packages. Okay, these are some of the packages that you can install. So I'll upload it to my GitHub channel, and I will provide the link in the description. Okay, so let's begin the notebook, where what you have to do is you have to load the dataset from the Visium, as I have shown previously, from where you get the dataset. And then you pre-process the dataset and normalize it using the Seurat v3 flavor, because Seurat v3 flavor is much more advanced to identify highly variable genes. Then you perform the PCA, the neighbors, enrichment, UMAP, and then laid-in clustering. Then you plot some of the static spatial plots, and then we'll be doing the interactive plots with the Plotly and Napari. So this is from the 10x Genomics dataset, so one of the demo dataset or example dataset from 10x Genomics. But if you want to load your own dataset, then you can visit to this 10x Genomics dataset. You get these files such as filtered feature bc matrix.h5, and then there will be a special folder as well. Okay, so you can get those two files and load it as data, Visium breast cancer or lung or brain, whatever you are trying to analyze. Pass all the data sets into a variable called adata, then you can load your data set, and you will be using the Scanpy package. From the gene expression omnibus, you have to get the H580 file or the matrix file, which is MTX files, and then you can simply load this by using the Scanpy package, and the command to load the packages is sc.read h580. Okay, or the MTX is path to the folder, and the variable names you have to set as gene symbols. If you have a CSV matrix like count matrix.csv, and you can convert the CSV matrix to n data by using this command. Okay, and then there you have to provide this particular package. I have listed all the packages, so you don't need to worry about the packages. This particular tutorial, what are the packages that are necessary, such as Scanpy, Squidpy, Plotly, Napari, with all the packages from Napari, scikit image, and data, matplotlib, and pandas. These are the packages that we require in order to run this particular notebook. So first thing first, we are going to import the libraries, Scanpy, Squidpy, matplotlib, pandas, and numpy, and then we can print the versions of all these packages. So I'm going to run this, and you can see all the version has been loaded, and the same exact file you find it over here with the exact version number, so that you don't have to worry about version mismatch. You get all the things over here, and you can see all the version, the dependencies, and what are the packages, etc. Move on to load the dataset. The dataset will be using its 10x Genomics Visium mouse brain dataset as an example, and you can load it via adata, which is the variable, and then you load the packages by using the Squidpy packages. So the command is sq dataset Visium and your data. Okay, now you load this, and you can see that you have some of the variables over here, which are the observation into the variables. Variables are nothing but your genes, observations are your samples. So we have like 2688 samples with 18078 genes. And then we go for the pre-processing of the data, where you normalize the data, log transform it, select highly variable genes by using this particular Surat v3 flavor. And top genes I am selecting as 2000 genes, 2000 highly variable genes. Okay, so you normalize it and then you scale and you find the highly variable genes over here. Okay, so once you do that, then we move on to the PCA and the UMAP and clustering. So once you load this, you can see that we have a number of components, which is the PCA components is 50, and the neighbors we are choosing to 10. So, and then you have the resolution here set as 0.5, which is I think more than enough to get a good picture of your tissue distribution or the cluster distribution. So we found 14 clusters and added them as cluster levels, and now you visualize the UMAP. So now you can see here, starting from 0 to 13, we have 14 clusters with different colors over here. And then for instance, if I want to find out a particular gene into the spatial plot, the plot that I have shown previously, the data set, if I have to put it that particular gene into this particular plot, then how it would look. So that's why we are looking for the static spatial plots over here, and I'm taking this particular gene which is called TCEA1. So you can see this is your spatial image. And then if you want to plot the TCEA1 over here, you can't see it visually by using this particular image, but you can also identify them by using some of the interactive plots like Plotly and Napari. Okay, these are some of the static images, by the way. And you can change the size also over here. So let's make it to 5.0. Let's see how it looks. Yeah, it's not yet visible because these are the static plots. So if I change it to 40, let's see if we can see the spots over there. Oops, enable scrolling. So there might be something over here, as you can see. You see these spots? Okay, these are the expression of the TCEA1. So that's why the static plots might not be that much clearly visible to the naked eye, and you can see these spots over here, the expression of TCEA1. Okay, so you can't visualize that, but that's why we go for the interactive plots. Okay, now we go for the neighborhood enrichment, so that we find out the cells which are clustered together and what type of activity they make. So you do that. Okay, and then you observe the clusters over here. It's finished. It's really fast because I have a small data set. And then you compute the clusters by using this neighborhood spatial neighbors function from the Squidpy package, and then you re-cluster them by using the Scanpy latent clustering algorithm, and you can see that now it's done. So this is how your cluster based on the neighborhood enrichment, like what type of cells activity, how much they are similar to each other, what type of cell activity is similar to what type of clusters. Okay, this is what the neighborhood enrichment is all about. Okay, now we can print the total number of spots. This is the spots, which is 2688. These are the unique clusterings, 14 clusters we found out, and you can see the counts per the cluster. So cluster 0 has 540 counts, 1 has 323, likewise. Okay, And then you can print out the first 20 genes and the last 20 genes, and you can see here these are the first 20 genes and these are the last 20 genes. And now you move on to the plotly, where you can take out any sort of gene. So I'm taking this particular gene, that's the gene, and I'm going to plot using the plotly. So let's see. Now you can see the plots along with the dots. So this is the expression levels, and you can see these dots, these yellow dots, this is the red dot, okay, and these are the expression of this particular gene in the whole spatial tissue, okay. And you can see these spots which has the highest expression levels as well. So that's why it is much more beneficial to plot it interactively rather than the static images, because you can't see exactly where it expresses, okay. So these are the spots where the highest expressions you can get, and this is like the blue ones are the like zero expression. You don't have any expression over here, only the red spot, the yellow spot, etc. you have. So you can also plot another gene. So let's take another gene such as sulf1, okay. So I'm going to plot this one first, and then I take in another example over here, okay, and then we plot that one. So you can see the sulf1 expression like this, and you can see this is the neural 3 gene expression, this is the sulf1. So you can plot many different types of genes of interest that you have. Let's say if we want to plot something like maybe this one, we can take col3a1 and we want to plot that one. So yeah, this is the expression of col3a1, this is the expression of sulf1, and this is the expression of neural 3. So likewise you can plot the genes of interest from here. And then we move on to this Napari, which is a desktop application, and you can actually plot that. So let's try this one with the neural 3 expression, and this is how your clusters looks like into the spatial image, and these are the expression of neural 3 gene expressions, okay. The plotly is much more beneficial. I think this is much more beneficial than the static image. I think these are for the publication purposes, but yeah, you can visualize the data over here, okay. Let's see if we can run the Napari over here and then try to implement it, because this is another desktop application. As you can see over here, this is a Python desktop application. So this is the Leiden clustering, and this is the neural 3. So let me just make it big, okay. And then you can plot them by using different colors. So let me plot them by using this color yellow, and Leiden clusterings I plot them as black, for instance. So yeah, you can play around, and you can see these yellow dots are nothing but your neural 3 expressions, okay, the yellow dots, and all the other dots are the clustering algorithms. So you can play around with this particular Napari visualization, which is much more beneficial to analyze interactively. There is also the plot called or the software called Loop from the 10x Genomics, that also you can explore. But I don't know much about Napari visualization that much. I haven't explored it yet, but you can explore this, and let me know in the comments then how you analyze, how you plot all the expression of the genes over here, and you can visualize all together, okay. This is kind of useful, so you can interactively visualize. But more than Napari, I like the Plotly kind of thing, which is even more you can identify what type of clusters they are, what is the expression levels, etc. from here. So yeah, that's it. I mean, this is how you run a spatial transcriptomics data, and you run your analysis by using your own data set or from some of the data sets from 10x Genomics. Okay, so if you like my videos, then please do comment, subscribe, and like my videos, and please do also suggest me what other videos that you will be interested in, so that I will try to make those videos for you. Thank you very much.